Here is the video on something we've been doing in class, but I want to make sure we actually see it and are able to take notes on it as well. So the question is, how do polymers form? If you remember, I've been introducing monomers, sugars, nucleotides, fatty acids, glycerol, amino acids, right? And all of these form these macromolecules, these polymers. Now the one we actually did during the lab, we took this molecule, polyvinyl alcohol, which you see it has a nonpolar hydrophobic side. It actually has two nonpolar hydrophobic sides. It's nonpolar because carbons and hydrogens are nonpolar bonds. So we have that here, we have that here. Nonpolar, meaning that there are no charges on the molecule, and then hydrophobic, meaning this part does not like to dissolve in water. And what we also have is this hydrophilic group. If you remember, oxygen is extremely electronegative, which means it pulls on electrons very hard. So this oxygen, being electronegative, pulls the hydrogen's electron a little bit more towards the oxygen, which gives this that partly minus charge and partly plus charge. This is exactly what water does. This is why water, if water molecules see this part of the molecule, they'll actually try to stick on with the water. That's what makes it hydrophobic. Hydrophobic, water-loving, hydrophilic. Or maybe I said that wrong. This is hydrophilic. Hydrophilic is water-loving. The minus here, the plus here, the partially positive and negative charges means that water will stick to that quite well. The hydrophobic portion, which has carbons and hydrogens, does not have those charges because carbon is not as hydrophobic. Carbon is not as electronegative as hydrogen, which means that carbon will not pull on the electrons as hard. So carbon and hydrogen share the electrons equally, which means there's no overall charge on this part of the molecule. This part will not mix with water. Carbons and hydrogens do not particularly like to mix with water. So if this actually dissolves in water, which you did actually see the molecule dissolving, these parts are going to be on the outside of my glue. And this part is actually going to stick to another molecule which has these carbons and hydrogens. Then you'd have these OH groups is on the outside again. So that's going to be one shape that this molecule can assume. If you noticed, the glue does still dissolve in the water, and that's thanks to these hydrophilic groups, which are on the outside while the hydrophobic is pushed towards the inside. Now the other substance we're using is sodium tetraborate. Tetra is 4, so this has 4 boron atoms in the crystal. What we did in this particular case, we linked together these monomers, which were polyvinyl alcohol, and we did that by putting these borax molecules. Now the structure of borax changes when it gets into water. When we dissolved it in water, the structure changes. But notice that these OHs, which are the hydrophilic portions, the part that like water, they're actually now getting linked on the inside of the overall molecule, making the hydrophobic portions stick on the outside. What does that mean? It means that now we've completely flipped around this molecule. Not only have we joined together these monomers, but we've also put the hydrophobic side on the outside, which means that this does not dissolve in water nearly as well. And as you were making the slime, I'm sure you noticed that the hydrophobic portions were on the outside, while the hydrophilic had been switched, right? Originally, this dissolved pretty well in water, made a nice cloudy solution, which was made of these molecules, the hydrophilic sides on the outside, the hydrophobic side on the inside of a mass of molecules where we had another hydrophobic portion here, another hydrophilic portion there. Here we've completely flipped it. We've pulled the hydrophilic portions on the inside and put the hydrophobic portions on the outside, and we've tied these together, which means these molecules do not dissolve in water anymore. We made a polymer. We definitely changed the properties of the molecules as well. That's where we made our slime. Now, we did not have any food coloring, but you get the right idea. That's what slime is. It's basically polyvinyl alcohol, which is mixed with borax to make it stick, and the hydrophobic portions are turned to the outside, so it doesn't dissolve in water anymore, and it does not dissolve in water anymore, and then you have your polymer. Now, the ones in nature are all linked together, not with borax, but with another process, which we call dehydration synthesis. Now, I've been trying to emphasize this in class as well, but that's basically condensation. We're making water come out of the air would be condensation. We're making water come out of these molecules. Therefore, that's the dehydration part. When we take out that water, we're actually putting monomers together. So we're linking these monomers. We're building a bigger molecule. Synthesis is building. Condensation means we're forming water, and we're forming our polymers by taking those monomers, taking out an oxygen and two hydrogens, and making a water molecule. So the basic structure, all of our monomers, this could be a sugar, this could be an amino acid, this could be a nucleotide, this could be a fatty acid. In every single case, on one molecule we have an H, on another molecule we have a hydrogen and an oxygen. You pop off that H and this H and the oxygen, and you get H2O. Now I'm realizing right now there was something else I needed to do. So I will have to go through this and edit this part out. Okay, now from there, once I have this completed, let's look at some actual specific monomers. These are glucose. These are sugars. When we put these together, we're going to make a complex carbohydrate. This is a simple carbohydrate. All of these angles are where a carbon should be, except here that's an oxygen. Carbon, 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 carbon. This molecule is called glucose. It's a 6-carbon sugar. What we're going to do is we're going to pop off a hydrogen and oxygen from one molecule and a hydrogen from another. We link them together. We remove the water. And what do we have left? So I take this off, these two off, I take this off. I'm left with an oxygen there. Whoops. I'm left with an oxygen here. That's the oxygen that now makes the link between one sugar and another to make a more complex carbohydrate. In lipids, now we also did this. This was the first activity we did, where we have the hydrogen and oxygen from the glycerol, and then we have the oxygen that's part of the fatty acid, and we pull off a water, pull off a water, pull off a water. Pulling off those three waters, now we have our triglyceride, which is another important thing to remember. In proteins, same exact thing. We take from the carbon side of an amino acid, and all amino acids are nitrogen, carbon, carbon. Do not forget that. This carbon is double bonded to an oxygen. Do not forget that. And then we have an OH here. This is what we call a carboxylic acid. And this is what we call an amine group. So we have our amino acid. You're pulling off a hydrogen and oxygen. You're linking it with another hydrogen. That comes off as water. And then we have two amino acids linked together. This R means that it's a variable group. We see this in amino acids. There could be a hydrogen there. It could be a sulfur-containing link there. Lots of things that you could see. Nucleic acids, again, we started to do this in class. On one sugar, this is what a nucleic acid is. It's a sugar, a phosphate group, and what we call a nitrogenous base. Nitrogenous base contains nitrogen. So on this 5-carbon sugar, not a 6-carbon like glucose, but this 5-carbon sugar, we have the OH, and then we have an extra H on this phosphate. We're going to pull off an HOH, and we're going to link them together. This is how we link our RNA. This is how we link our DNA, and then we have a water. And the way that macromolecules are separated is actually identical to, well, it's the reverse of the way that they're put together. So in this particular case, we have hydrolysis, water breaking, hydrolysis. Also digestion. This is what living things do to break down big polymers into their monomers. What do they do with those monomers, like a sugar or an amino acid or a nucleotide or a fatty acid? They use that to build new and different polymers. So you break down the protein that's in a plant in your salad, you break it down to the amino acids, now you build human proteins out of those amino acid building blocks. The building blocks are the same between a plant and a human. However, the polymers are going to be different. So in carbohydrates, you have two glucose molecules linked together here. You add in a water and they break apart. You take that water, one oxygen goes to one molecule along with a hydrogen, and then a hydrogen goes to the other molecule. In lipids, you see the same thing. Here's the bond that we built. Now we put it in a water, we break that bond. This O should be also orange, but this is the break, so H2O, same thing. In the proteins, again, I'm putting in a water, the OH ends up on one of these amino acids, and the H ends up on the other. Now this is a peptide, meaning this is still connected to other amino acids. A peptide is a chain of amino acids. We don't know what those others are, but this is one amino acid that has come off of my protein chain. And the nucleic acids, again, the same thing. We're breaking apart these nucleotides. This is the nitrogenous base, the 5-carbon sugar, and the phosphate. We're breaking this by adding water. So that's the important thing to understand about dehydration synthesis and hydrolysis. Dehydration synthesis, let me get back to that slide. Dehydration synthesis, do not forget synthesis, building. Dehydration, taking out water or condensation. And then hydrolysis, hydro, water. Lysis, breaking. We're breaking things apart by adding water molecules. Very important to remember. Thank you, and I'll continue with the importance of these macromolecules in the next video.